sir. Good morning to one and all present. Welcome to Dr. Dwaipain Goswami. Before I take pleasure of introducing our webinar speaker, please allow me to express my gratitude to Dr. Dwaipain for accepting our request to deliver a webinar for Parul University. Thank you so much, sir. Sir is going to deliver a webinar on map of immune system. He is professor at Department of Microbiology and Biotechnology at School of Life Sciences, Gujarat University. He has a teaching experience of about 5 years. He has cleared CSIR net exam overall 30th rank. Sir is having over 30 plus research papers and over 700 sites of 10. He is having expertise in computational biology. He has also published 3 research papers on corona during this pandemic era. It's a long list, students. So, without wasting any time now, I would like to invite Sir to proceed with the webinar. And also, I would be liking, if you have any queries, you can directly message me in the chat box. Sir, it's all up to you now.
Thank you, Dr. Farad, for just giving wonderful explanation or introducing me to the students. I first question, why I would like to give this in the era when each and everyone in this scientific fraternity is talking about coronavirus. Number one. Number two, why this? This is because immunology is such a vast subject that if I have been doing regarding in this world, how vaccinations are working to help basic to insane when to the student. And after teaching this subject again and again and again and again for 4 to 5 years, I just came across the fundamentals that there is a fundamental rule to understand immunology. To whom I'm addressing, probably I'm addressing to the students of BSc and MSc, I guess. Okay, so there are different bits and pieces of immunology that is there in the books. There is bits and pieces of immunology which is there in the topics or the syllabus that we are studying. This is not the fault of the system. Okay, this is basically to understand and defragment the entire subject and understanding bits and pieces of the subject. Okay, so now the scenario is such that let me give an example. Suppose you know how the tire works, how the tire looks like, you know how the engine looks like, you know how the headlights of a car looks like, but probably you don't know how the actual car, when you assemble the things together, how does it look like and how does it function. Okay, so just coming to the topic, I just want to tell you that suppose you would have understood the terms like humoral immunity, cell mediated immunity. Okay, then you have acquired immunity. Okay, innate immunity. And then there are something which is known as human leukocyte antigen, HLA, MHCs. Then you have complement systems. Then you have antibodies. So how does all these things, the different pieces of the entire immune system, will come together and work as a function? Okay. Now, when you are trying to understand immunology, you just need to understand that there is a fundamental rule and the fundamental law, that a fundamental branch of immunology that first of all you need to understand. Okay. Once you understand that branch of immunology, the other factors come in and it will make the subject as whole. And when you have the entire subject as a whole, and when you, it is like a Google map. So when you zoom a particular part of immunology, you have more and more information coming out of it. Okay, so I will try to make this webinar as interesting as possible without boring you, because I know generally how the trend is in the today's era, where you know there are so many renowned personnels will come to you, they will talk about their research, Okay, and you will find it very fascinating for first 20-30 minutes, and then probably the the energy boosh, it keeps on going down and down. So we don't want that to happen. We want to have a fundamental understanding about the subject that will stay with you for a pretty long time, and that will help you to understand the other branches of immunology, actually come across these topics. Okay, so without wasting any time, I will just present my screen. If there is any problem with my screen sharing, please, Dr. Farad, please let me know.
Sure, sir. Sure.
So I have just so now can you see my screen? Is my screen visible? If yes, then I will proceed.
Yes, sir.
Okay, okay. So let us just talk about the immune system and the immunology. Okay. So before we move further, the immune system works at a different places, at a different time, at a different stages. Now, what are the stages? So immune system works when you are having a bacterial infection. Immune system works when you are having a viral infection. Immune system works when you are producing, your body is producing cancer cells. Immune system works when you are having some injury in the body. Okay, even the healing of bones, healing of skin, healing of different components of the body, the organs, the tissues, everything is pertaining to immunology. Okay, so now to understand the basic fundamentals, we will start by understanding how the immune system, the basic underlying principle of immune system will work when there is an infection. Okay, and we will start with the bacterial infection. Why? Because how, when there is a bacterial infection, how exactly the immune system works, the entire hierarchy is well defined, okay, and understood. Okay, so just assume that there is a tissue, any tissue of your body. It can be a skin tissue, it can be a tonsil, it not a tonsil, but it can be any tissue, any epidermal layer of your skin, body, whatever. Okay, now assume that the bacteria, okay, after crossing the first line of defense. So now, first of all, what is the first line of defense? First line of defense is nothing but the physical barriers. Okay, so your bacteria or the pathogen is crossing the first line of defense, crossing the barrier of physiological boundaries of your body, is somehow able to get entry inside the tissue. Okay, so just assume that your finger is been cut or it has been wounded and a bacteria is entering inside the finger. Okay, the skin of the finger. Okay, so now let us just assume the bacteria is entering into the tissue of your skin inside finger. Okay, so the currently this bacteria, for my understanding, and I have some particular love regarding bacillus, so I have kept the bacillus over here. So your finger has been wounded and there is a bacterial infection with some bacilli, okay, inside the tissue of the finger. Okay, so now what is the first thing that will happen? Okay, the first thing, or the multiple things will happen at this place. Okay, there will be inflammation. Okay, then that is the first thing that will happen, inflammation. Okay, but let us keep that aside. Let us understand the fundamental thing that is going to happen, and how your body is trying to how your body is, how does it deal with this infection. Okay, now when I'm drawing a single bacteria, doesn't mean that there is only a single bacteria. Okay, you just need to know that I have drawn a single bacteria to just make things clear. Okay, so there cannot be a single bacteria causing infection. There has to be a particular threshold concentration of bacteria in order to cause some sort of infection. Okay, so for your just basic understanding, I have kept one single bacteria. Okay, but in reality there are more than uh, 10, 15 or 100. What is the first thing that will happen? Okay, so over here, this is what I have drawn is the blood. Okay, so in the blood there are two major okay, and these two players will come from WBC. Okay, and those players are monocyte and neutrophils. Okay, now these are the key things. These are the cells which comes first. Okay, so monocyte and neutrophil, they both belong to the category. Leuco means WBCs. So monocyte and neutrophils, they both are different types of WBCs. Okay, Now, they both are there right now inside the blood. Okay. Now, what is this? Means we are trying to see two different stories and then we will club them together. Okay. So just assume that there is a war going on some border of a country. Okay. The personals who are standing on the land, okay, they have different weapons. Okay, the air force is having a different weapons. Okay, and both of them some way together they are trying to kill the opponents. Okay, so that is the basic understanding. Okay, so first the entire story we will see how the macrophage or a monocyte is going to work, and then we will see about the neutrophil. So right now we are keeping a neutrophil aside. Okay, so this neutrophil will first enter inside the tissue. Why? Because neutrophil is the smallest in size. Out of all the WBCs that you have, neutrophil are very, very, very small. Okay. And neutrophil from the blood, it will enter into the tissue which is infected with a bacteria. Second thing, the number of neutrophils in the blood is maximum. So compared to WBCs, neutrophils are very, very, very large in numbers. So the neutrophil will enter inside. After neutrophil, macrophages will enter. Okay. Now there are two different stories. Let me just go back. Macrophage and neutrophil, there are two different stories. Okay. So first we will understand how macrophage is going to deal with the infection, and then we will understand how the neutrophil is going to deal with the infection. We are trying to understand one after the another, but in reality both the processes are occurring simultaneously. Okay. Now the process, so the process that I have been showing you over here, that the neutrophil from the blood, it is entering into the tissue. Macrophage from the blood, it is entering inside, sorry, a monocyte is entering inside a tissue and it is becoming a macrophage. Okay, this process is has three different words: leukocyte migration, diapedesis, and extravasation. So there are three different terms from the same phenomena. Okay, now as soon as, if you see, just go back. Now what is the name that I've shown you over here? Monocyte. Okay, so monocyte, when it is in the blood, we call it as a monocyte. After it enters inside a tissue, it becomes a macrophage. So monocyte and macrophage, they are a same cell. You just need to remove any confusion if you have regarding this. Monocyte, when it is circulating in the blood, monocyte, when it enters into a tissue, it becomes a macrophage. Okay. So now let us just see the phenomena, what is happening with a macrophage. Okay. So now this is something that we want to know. Now macrophage, when you break the term, it is a macro, which is large in size, and phage means a cell which has the ability to do a phagocytosis. Okay, so phagocytosis is very basic term that you have understood. The first thing that will happen is that this macrophage will create a pseudopodia around itself. Very simple process, probably everyone would be knowing about this. It will engulf the bacteria. Okay, and the engulfed part around this white ball that you can see with the bacteria is known as the phagosome. It is a part of the phagocytosis process. So the phagosome is created. Okay, now we will just enlarge the image and then let us see what is happening. Why this is very important? Because ultimate goal is to kill the bacteria. We want to kill this bacteria. So in order to kill this bacteria, there are three processes that this macrophage will do. What are these three processes? Oxygen dependent killing of microbe, respiratory burst, and oxygen independent killing of microbes. So ultimately, with the help of oxygen and without the help of oxygen, we are going to kill this bacteria. So if you want to go into details how this exact process is going to take place, okay, then you go to any reference book and understand what is oxygen dependent killing of microbe. Okay, but just for the understanding, this is just a map, so I will not go into the details. Just assume that we are going to kill this bacteria with by hook or by crook. Okay, now inside the macrophage there are these vesicles, these wonderful small vesicles that are containing digestive enzymes, and these small vesicles are known as lysosomes. Okay, so lysosomes, phagosome, and it will known as a phagolysosome. So this process that you are seeing is a part of oxygen independent killing of microbe. Here we are not, just for the ref, in the basic fundamental hierarchy, we don't want to incorporate this right now. Okay. Then, so the lysosomes will secrete different types of digestive enzymes, and it will try to degrade the bacteria. So ultimately, we are killing one. Process is very slow. Okay, but this is the major checkpoint occurring in the immune system. Why this is a basic and very important uh, checkpoint in the immune system? Just a second, I will tell you. Because as soon as this process is taking place, as soon as the bacteria is degrading inside the macrophage, a signal is given to the chromosome number 6 of macrophage. Now this is something which is very important. Okay, the chromosome number 6 of the macrophage has the portion which is known as HLA. So inside chromosome 6 there is a region which is known as HLA, and that is human leukocyte antigen. Okay, now someone knows, this chromosome 6 knows that, okay, fine, there is some issue in the system, so let me get activated and let me produce something. So this chromosome number 6 produces these wonderful molecules which are known as MHC class 2. Okay, it is known as major histocompatibility complex number 2. Major histocompatibility number 2. So again, very simple process. Chromosome 6, it will start producing certain mRNAs. It will become a protein. So from the information from the mRNA will go to ribosomes. Ribosome will make a protein out of it. That protein will become mature in the Golgi bodies, and from the Golgi bodies, after MHCs are getting matured, it is in the form of a vesicle that you are seeing over here. Now this vesicle containing MHC will fuse with the phagolysosome, and the wonderful process takes place. The small components of the degraded bacteria, which we call, what do we call this? These small components of a degraded bacteria we call as an antigens. So these small antigens are getting loaded on the MHC class 2 molecules. Okay. As soon as they are loaded on the MHC class 2 molecules, this MHC class 2 molecules containing this small antigens of the bacteria is exposed outside. Okay, what is the purpose of exposing this antigens on the outside? Okay, so now let me keep everything clear. Suppose you are sitting in the you are living in a very large palace. I'm talking about 100 rooms. Okay, you have 5 different kitchens, and there is a big palace. Okay, so if a thief is entering into a palace, what will happen? Somebody has to tell that there is a thief in the palace. Okay, so the chowkidar or the guard which is there on the gate of a palace screams, okay, runs a bell or rings a bell to tell the entire audience inside the palace that there is a thief inside the palace and we need to take some action against it. Okay, so the exact same procedure has been done by the macrophage. Okay, this macrophage is trying to show that there is the problem in the body and there is the antigen which we have to deal with. There is a bacteria that we have to deal with, and this macrophage, like a news channel, okay, it will start running the news throughout the entire body that, okay, fine, there is a problem in the human body, we have to do something about it. Okay, so the basic activation of immune system starts from this point onward. Okay, so just remember that this process is taking place again inside the tissue where the infection is taking place. Okay, once this process is done, this macrophage will again go back to the circulatory system. So it is again going back from the tissue of infection to the circulation. Okay, then it has to go somewhere. So where will it go? So from this circulation, it will go to a very specialized place which is known as a lymph node. So this can be a lymph node, this can be spleen, or this can be any other secondary lymphoid organ. So the process is going to be the same. Okay, so it will just go over to the lymph node or to spleen or to any other secondary lymphoid organ. It will just stay over there. Okay, and it is trying to show these antigens to the entire immune system that we have the thief in our body. Okay, so now what is going to happen? Let us see. Okay, so this macrophage, okay, will go inside the lymph node, and inside the lymph node there are these wonderful cells which are known as the T helper cells. Okay, now why they are called helpers, you need to know. I will give you the reason later on, but they are there. Now what is the unique thing that you could see over here in this image? The unique thing that you see in this image, that despite this T helper cells being of a same community, they are being the same cells, they have these molecules which are in different colors. So here it is a white color, then there is a green color, yellow color, red color, blue color. So what does this color indicate? This color indicate that there is one unique protein outside this T helper cell which is working as a receptor, and each receptor is unique. Okay, this receptor is unique. Okay, now what is the basic fundamental that we are trying to see? The basic fundamental is that this macrophage is trying to show the and to MHC is just a wrapper of the gift, and the gift is inside the MHC molecule, which is this yellow dots. So this is MHC class 2 molecule, which is a gift, and this to this T helper cells that see, please, we have a problem in the human body. We have the problem. Please, 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 I'm pleasing you. Please get activated. So this message is given by the macrophage to the T helper cells. Okay. Now, just for a few instances, let us see what is this T helper cells. Okay. So now just we are zooming in just one of the T helper cell, and just we are trying to see what they are. Okay, so the yellow molecule which you are seeing over here is known as a CD4. So don't go into the details right now, just remember it is known as CD4. Okay, and the two chains, the two antennas that you see on the top of the T helper cell, you call them as the alpha chain and the beta chain, and alpha and beta together they are known as a T cell receptor. So this entire molecule is known as a receptor. Okay, now there are certain details that I will explain when we will see what is happening. So all these T helper cells, they are twin brothers or sisters. You can call them whatever you want, but all these T helper cells, okay, are having this different antennas on them. Okay, now you just remember all these antennas may not be able to identify the antigen. Okay, so now let us see what is happening. Only one out of the several cells can identify the antigen which is loaded on MHC class 2. Okay, only one out of these hundreds and hundreds and hundreds of cells which are there in the lymph node, only one cell, just remember, only one T helper cell can identify the antigen as a foreign antigen. Okay, and it can go over there and it is trying to receive the gift from this macrophage, that okay, fine, let me understand how this antigen is. Okay, so now this is what is happening. So now this is T helper cells. So T helper cells are very difficult to please. They are not someone which you can please every time. Okay, they are very, very, very cautious. Okay, they are they are the cells, you can personify them, the cells, as the cells with the overblown ego. They are the kings and the queens of Hollywood and Bollywood. Certain kinds of ego they carry. Certain kind of ego is been carried by the T helper cells. In order to activate this T helper cell is the first phenomena. If this T helper cell does not get activated, then the immune system is not getting activated, friends. And in order to satisfy the ego of this T helper cell, this macrophage is trying to do so many different things. And once this T helper cell agrees to the entire thing, finally the T helper cell will get activated, and then the entire immune system is getting activated. So this is the entire picture. But there is certain amount of effort to be done by the macrophage in order to satisfy the ego or the satisfaction of this T helper cell. Now, each of this receptor is different. Therefore, only one of the T helper cell with a unique receptor affinity for the antigen is selected, okay, for the activation. And this process is known as a clonal selection. Okay, so now if you want to go into the details, look into the QB, what is exactly clonal selection. Now you have the better understanding of what you are trying to, what you are expecting from the topic, what you are expecting from this from this process. Okay, so right now this cell with this yellow alpha and yellow beta is identifying this antigen which is holded by the MHC class 2 as a foreign. Okay, now once this lock and key, it is like a lock and key, so when this lock and key is taking place, what is happening? Okay, this T helper cell is doing a lot of nakra. So I'm not going to get pleased so easily. Even if this antigen is foreign, I'm not going to help you out, dude, please. So I need more. So now what is happening? uh, okay, macro on the surface, B7 will bind with one more molecule on the surface, which is on a CD24. So one more lock and key is taking place. So just assume that this macrophage is trying to please the T helper cell. Okay, and as soon as this lock and key is done, okay, the signal is going inside. The signal is going inside the nucleus of a T helper cell, and as soon as the signal goes inside, this T helper cell start expressing this yellow color molecule on the outside. And this yellow color, red color, this red color molecule is known as the interleukin receptor. Okay, now see just the amount of how fuzzy this T helper cell is. Okay, now what is happening? This macrophage is going to secrete some molecules which are known as interleukin 1. So interleukin 1 will go and it will again bind to the receptor of T helper cell, which is a interleukin 1 receptor. Again a signal is going inside. Okay, all this signal is trying to activate this T helper cell. Okay, that go, oh please, 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 please try to get activated, we have a problem. Okay, but this T helper cell is doing a lot of nakra. Getting, give me enough amount of signals so that I get activated. So 3 signals we have received till now. Okay, one from T helper, one from the first signal is coming from this interaction of MHC class 2 and T cell receptor. The second signal, which is B7 and CD28. Third signal, interleukin 1 receptor. Now after this 3 signal, again the T helper cell is trying to secrete or produce another type of receptors on its surface. Those are known as interleukin 2. And therefore this part 2 receptors on going. Okay, and this process is known as autocrine signaling. So if you see over here, first interleukin 1 interaction is a paracrine signaling, interacting with a receptor and a signal. And over here, if you just go back, it is a autocrine signaling. Okay, so interleukin 2 autocrine, interleukin 1 paracrine. So this signal is there. So just assume how many different signals this T cell is receiving. Okay, so this T cell is receiving 1, 2, 3, and 4. At least 4 different signals this T cell is receiving. And as soon as there are enough signals received by this T helper cell, okay, so then now T helper cell is satisfied that okay, fine, I am convinced that there is a problem in the immune system, I have to get activated. Now at this point, it is another checkpoint that the immune system is getting activated. Okay, and now the T helper cell, after receiving this 4 different signal, it is known as a activated T helper cell. Okay, till now it was not called as activated, it was just a T cell. Okay, but once it receives these 3 different signals, it is called as a activated T helper cells. Now once this T helper, what is happening, let us just see. They are trying to proliferate, so they will simply multiply. But now what is the beauty? The beauty is that when this cell is multiplied cells have the same of, if you see in the previous case, they were all different types of receptors on the top. So now only the activated cell is now multiplying or trying to multiply. Okay, and when it is multiplying, okay, when the cell multiplies at this point, it is trying to produce only a same type of receptors, and this process is known as a clonal proliferation. This process is known as clonal proliferation. Why? Because they are all clones of one another. Okay. Now they are activated. So now just you understand. First macrophage only knew that okay fine there is a problem. Now also T helper cells know that there is a problem, and as soon as the T helper are activated, they are trying to multiply at such a fast rate in your body that if you take the blood, okay, and if you just try to see the blood report, if you do the blood report directly, you will see that the amount of WBCs in your blood will shoot up. Okay, that shooting up of WBCs is because of some part of it is because of T helper cells multiplying at a very fast rate. So now from one cells, millions and millions identical T helper cells are getting formed, and all these cells which are multiplied at this stage all are activated. Okay, and these activated cells are producing these molecules which are known as interleukin 4. Okay, so we have understood interleukin 1, interleukin 2, interleukin 4. So now where is number 3? We'll just see. So this is producing interleukin 4. It is also producing another molecule interleukin 5, and which is invisible over here, it is also producing interleukin 3. So let us first talk about the ones which is invisible right now. I have not shown you over here. The T helper cells also produce interleukin 3. So interleukin 3 are the signals. These are the signaling molecules, interleukin 3. They will go to the bone marrow. And in the bone marrow, there are a lot of cells which are known as stem cells. Okay, so interleukin 3 will go to the bone marrow. They will give the signals to the bone marrow that, okay, fine, we have a problem in the body. We need the bone marrow to produce more number of WBCs to fight with. Okay, so it is the molecule, when it reaches the bone marrow, it will tell the stem cells to produce more and more and more number of WBCs to fight against the infection. So interleukin 3 I have not shown because it is not a part of the central hierarchy that we are studying. Okay, so till now we have seen interleukin 1 produced by macrophage. Okay, interleukin 2 produced by not activate non-activated T helper cell. Interleukin 3, interleukin 4, interleukin 5, all these three interleukins are produced by T helper cells, and as they are producing only and only interleukins, they are not producing anything else, friends. They are not doing anything to the bacteria. T helper cell is doing nothing, but they are only producing interleukin 3, interleukin 4, and interleukin 5, and all these signals is going to activate the entire immune system and other cells of immune system, and therefore they are called helpers, because they are helping the entire immune system to get activated by basically three different interleukins: interleukin 3, 4, and 5. Okay, so this is just a half of the story. Okay, macrophage. Let us go just see and with the new. Let us see what is this neutrophil trying. For all clear till the macrophage. The macrophage story ends over here. We will start again with the neutrophil. Okay, this process is also taking place simultaneously. Now you need to remember, neutrophil is phagocyte, but they compared comparison large with neutrophil having two phagocytes. The large phagocyte that side the neutrophil process. Okay, it will travel this process parallel. You just need that previously, that oxygen dependent killing of microbes, respiratory burst, and oxygen independent killing of microbes. All these three processes also takes place in the neutrophil. Okay, so the neutrophil number is not simply bothered about what is happening outside. Unlike macrophage, you know, say okay, it was nothing else. I don't have any pleasure of pleasing the T helper cell. So no T helper cell coming into the picture right now. Okay, so this neutrophil is just digesting the bacteria. Piece then has been just it doesn't care about now. Lymph helpers again cells. Okay, these are there is a rule between B cell and T cell. What is the rule? These are very, very easy to please. Okay, nothing like thugs on the streets. So these B cells are immune system and they are thugs of the lymph node. So, okay, and they are simply waiting for one or the two different antigen, foreign antigen to come. They are just waiting for the antigens to come. Okay, till this point, B cells are sleeping. Okay, okay, assuming, but before what the T or B cell will look like. Okay, so this B cell, I'm just showing you over here a mature B cell. A mature B cell has a surface bound immunoglobulin. So I believe that even if you are in directly coming from the school, you know this, that there is something which is known as IgM or immunoglobulin. Okay, so Ig. Now, uh, just parallel, and there is this alpha and beta chain which is 29. Okay, so when in this B cell immunoglobulin or antibody together, they are known as a B cell receptor. Okay, and as you know very well, each antibody has a different variable region, and because of this different variable region, no two different from two different B cells are identical. Okay, I'm using the word no. Antibodies, immunoglobulins from two different cells are identical, so they have this point. Just let have given antibody showing. Okay, so let us see what else do we have. So we have membrane bound immunoglobulin. So this can be immunoglobulin M or D. Doesn't go into the immunoglobulins molecule which is together within the antigens. Okay, so now what will happen? Find with a unity and will again election is in the is everywhere for the B cell, for the T cells, anywhere. Now let us just club the story. B cells are very easy to access in receptor is D CD sent to the nucleus of the B cell, and F cell immediately is again, so there is one on now Introduce the right go and they will bind receptor are cell growth. Another name growth factor will go over there again. Cell but growth will happen. Division. Receive of the B activity. Okay, of a B cell. So this process is known as activation of a B cell. So if you open QB, this process I have just shown you over here is an entire chapter. Okay, so it is as deep, but we are just using the map of immune system, so I'm not going into the details. Now what is going to happen? These B cells will proliferate, and when this B cell will proliferate, they are known as clones of one another, and when they are clones of one another, they are having the same antibodies on the top. So active particles. Okay, when same with now the surface are known as receptors. In 5 and interleukin 5 will go and it will bind on this receptor, and what is the interleukin factor? We are trying cells that are multiply and they are cells. Some plasma interleukin 5 has to be much greater. Okay, now what is the difference between the plasma cell and the B cell? Simple. Plasma cell antibody on the outside, whereas the B cell have very few antibodies and they will indigenous. This if I antigens and antibody, please pardon me. So antibody, the word, very sorry. So antibody. So B cell has a surface bound antibody cell or with the okay, it is secreting the also secreting. Okay, but which antibodies? The same variable region which previously it had. The antigens is also a part of clonal protein taking place inside, which is known as a class switching. Why? Because initial membrane or types antibody A and okay, and immunoglobulin E, okay, plasma cell, the class is known as, whereas B cell was able to produce only IgM or IgD. Okay, so with the help of T helper cell, B cell could multiply. You just see, you just try to connect the two stories. Only with the help of T helper cells. If T helper cells would not have been there, B cells would not have been getting activated. Okay, because T cells is producing interleukin 4 and interleukin 5. Okay, and without interleukin 4 and interleukin 5, these B cells would not be getting activated, and therefore these T cells are known as T helper cells. Okay, now when we are proceeding further, this is the entire map of how this B cells are getting activated, T cells are getting activated. But there is some intricacies over here that I will try to address later on. So these antibodies that are there, which has been secreted by the plasma cells, they will circulate throughout the body, in the lymphatic system, in the circulatory system, everywhere. Okay, this will go to the place where the infection is. Okay, and pretty well know these antibodies are very much important to kill the bacteria. Okay, and this is the first step. This is not the first step, but this is the third checkpoint. Okay, so this entire process after infection, this will take around 7 to 8 days to occur. So if you are having a severe infection of a bacterial infection, okay, the maximum amount of antibodies in the blood would be detected after 7 days. So the process that I have shown you will take 7 days. Okay, days up the deactivate the bacteria. For 7 days, what is immune system doing till the 7 days? Okay, try to are trying to control the infection. Okay, come into or 5 days. If you are having a previous infection, okay, if there are memory cells already with this identical antibodies, 2 days. Okay, but we have to wait for the antibodies to get secreted to neutralize the infection. Till that point, the infection is not going to get cured. Okay, but that doesn't mean that immune system is not doing anything. Till 7 days, this neutrophil and macrophages are trying to control somehow this infection. Again, what is the function of these antibodies? So these antibodies, once they are released, okay, they are attaching to this bacteria. Now just assume that we all love pizza. Okay, but can you eat a pizza without cheese and without the tomato puree? If I just give you the pizza base and I will tell you, okay, fine, this is a pizza, eat it, will you eat it? Of course not. Okay, so we need to have a complete pizza in order to find it very tasty. Similarly, these macrophages and neutrophils would like to have this bacteria surrounded by this antibody, and only if this bacteria is surrounded by this antibody, it is becoming very tasty for the okay. And once, just remember, once this bacteria is surrounded by this antibodies, the rate of phagocytosis will increase up to 50 times. Okay, मतलब यहाँ तक neutrophils and macrophages जैसे तैसे करके it were trying to eat this bacteria, but it was not finding it tasty, so the process was very slow. But now the immune system is activated. Now the antibodies is trying to cover the entire surface of the the bacteria, and now this bacteria is neutralized, and once this bacteria is neutralized, the rate of phagocytosis will shoot up by 50%, not by 50%, 100 folds, up to 100 folds. And as the rate of phagocytosis will increase up to 100 folds, what will happen? The infection will be cleared. Okay, so this is the entire picture. This is the basic understanding of how antibodies are very important. Okay, then this is not the only task that has been done by the antibodies. Antibodies, first of all, it is going to make the bacteria very, very, very tasty for the macrophages and neutrophils to eat. Okay, and therefore this process is known as antibody dependent cell mediated toxicity, ADCC, and it is also known as opsonization. The term opsonization is nothing but you put the gravy and the tomato puree, which is also called as a gravy, tomato gravy, and the cheese on the top of a bacteria, so that this bacteria is becoming very tasty for the macrophages and neutrophil to just come and eat it. Okay. So this is also known as antibody dependent cell mediated toxicity. Why cell mediated? Because now the phagocytosis is going to take place very quickly. And phagocytosis is what? It is a cell. So cell mediated cytotoxicity. It is also called as opsonization. Why? Because the bacteria is neutralized. Okay, the bacteria is handcuffed. So therefore, it is neutralized. Okay, so again opsonization. Okay, again neutralization. Why? Because at this point bacteria is neutralized. And if you remember, if you cover the surface of a bacteria, what is there on the surface of a bacteria? The surface of the bacteria has a lot of lipopolysaccharides if it is a gram negative bacteria, and lipopolysaccharides are toxins to the body. Okay, just remember the mycobacterium, the membrane of a, okay, so it is very reactive. So the phagocytes like macrophage and neutrophil, they will think twice before eating that bacteria because of the toxic nature of lipid bilayer. But now, as this bacteria is neutralizing, this bacteria is neutralized, okay, the phagocytosis is becoming very quick and the infection is being cleared. Okay, so this was the overall picture of the immune system. Now let me go back to the first slide. So my explanation is over till this point, but there are some loopholes that I want to just fill in. Okay, so just let me go back to the first slide. Where is the first slide? Okay. Okay, so I have over here shown only two different type of cells. But if you want to just understand what this nature is, this monocyte could also be a dendritic cell. So dendritic cell and a macrophage. Okay, so when these cells are coming inside, I have just shown you two cells, neutrophils and macrophage. But remember, if I write a dendritic cell, the process is going to be the same. Okay, so if it is a dendritic cell, if it is a macrophage, the process is going to be the same. Okay, so what is the process? Phagocytosis. Okay, chromosome 6 getting activated. Okay, and it is MHC class 2 is showing this antigen on the outside. Okay, so entire this process which I've shown you over here, it is known as antigen processing and presentation. Why? Because it is presenting the antigens on the outside. Okay, so macrophage can do this task, dendritic cell can also do this task, and to your itself. So the B cells which identically when they are sleeping on the outside, so B cells, where are they? Let me just cells where will come over here. Okay, to your surprise, the B cells which are unactivated or non-activated B cells, the non-activated B cells are also doing the work very identical to macrophages and dendritic cells. So you can simply put this B cell over here on in place of macrophage, and they have ability to express MHC class 2 molecules. Okay, so okay, so okay, so this cell can be a macrophage, this cell can be a dendritic cell, and this cell even it can be a B cell. So if it is a B cell, it will have an antenna on the top. That's it. It will have an antibody on the top. That's it. Okay, and as they have ability to represent it MHC class 2, macrophage, dendritic cell, and B cell, these three cells are also called professional antigen presenting cell, because they have this ability to express MHC class 2 on the surface, because they are doing little bit extra after killing the bacteria. Okay, so I hope that this is the entire picture of my immune system is going to make some sense, and it will stay with you for a pretty long time. Okay, and if we are just talking parallelly, so macrophage is trying to eat this bacteria, neutrophil is trying to eat the same bacteria, dendritic cell is trying to eat the bacteria, B cell, even if it is there in the circulation, it is trying to eat this bacteria. Everything is trying to eat up this bacteria, but parallelly, at the same time, at the same moment, what is happening? There is something which is known as a complement system is also trying to kill the bacteria. So here the map is merging. We are just understanding a single aspect of it. Okay, so with this, I hope I am on time. I am exactly it is getting 11:25, so I hope I have done the justice to the topic and justice to the time. I can simply uh, hand it over to uh, Dr. Farat.
Yes, sir. Thank you so much, sir, for such an informative and in-depth webinar on map of immune system and actually getting us very clear to the basics of immunology. Really, really thankful to you.
Thank you. I would like to know the comments if there are of students, if they are
I,
viewing us.
yeah, so they'll let me know, and I'll definitely let you know as well soon.
Okay,
Actually,
thank
I'm
you,
really
thank you.
very grateful of you, sir, for taking time out from your busy schedule to share
Mhm.
your expertise with our students. And it was actually my
Thank you,
pleasure to share my workspace with you during my PhD journey. And I'm really happy that you have always helping me during my
thank you,
PhD
Farat.
journey,
Thank
sir.
you, Farat. Thank you, thank you. Same goes over here. A lot of gratitude from my side as well.
Okay, so what I'll do is I'll collect the questions, sir, and would direct that questions to you so that it would be
Sure.
helpful for the students as well to know the details. Thank you once again, sir. I also thank all the faculty members and students for their kind cooperation and doing being with there during this webinar. So hereby I conclude that the webinar hence is completed. Thank you
Thank
so
you.
much, sir.
Thank you. Thank you, Farat. It was nice
Bye,
having
sir.
you over here and me being a
Same
part
here,
of
sir.
this webinar sessions that you have. Thank you so much.
Yeah, my pleasure, sir.
Thank you.
Bye. Yeah.
Bye bye.